is non-random matings. Now, Hardy-Weinberg assumes that those don't happen, that basically is all random, that everything just happens and everybody mates with everybody else, and there's no preferences whatsoever. There are multiple forms to non-random mating. Some are really extreme compared to other ones. So for example, the first group is called assortative. Assortative includes things like ones that are based on phenotypes. So a particular female bear decides to only mate with other male bears that can push down a certain tree or find a certain kind of berry, or if it's birds, it can make a certain kind of nest. Maybe the female bird only wants males that can dance a certain way or produce a certain kind of nest. Those are phenotype ones that are based on very specific traits based upon your genetics and your learning abilities. Pair bonding is another one, because random matings don't have pair bondings. You mate with everybody. Pair bondings occurs in lots of animals, including things like humans, where people will basically pair up and mate for life. For some species, like penguins that do this, for example, emperor penguins definitely do this. They pair bond for life. A lot of bird species do this. In humans, it can be based on things like your religion. So maybe if you are Zoroastrian, Zoroastrians can only marry other Zoroastrians by the religion's decree. So that would become a concept like that. Or maybe you pick somebody based on the job that they have or the type of lifestyle that they live. So there's all kinds of traits like that that we utilize as our evaluation socially. So this is one of those things where you can see the effects of matings. In this case, another type of problem could be inbreeding. Inbreeding is a very specific type we'll talk about here. You can see where it can throw off the random mating concept because it's not following a traditional thing. It starts to throw off the overall concept. Because inbreeding is one of those types of things that becomes a problem. Mating between relatives or close related individuals. There's a coefficient of inbreeding that we can use to measure how bad or how close this concept is. That's the big letter F here. So it determines probably that two alleles at a locus come from a common ancestor. How likely is it that it comes from the common ancestor? The more likely it is, and the closer that number is, that means there's more of a chance like having a copy of a copy. It can become problematic because it can actually interfere with each other. It can actually end up becoming very much geared towards one allele over another. Brother-sister matings have an F value of 0.25. So that's, if you have a direct brother-sister mating, that's how that concept works. This occurs, obviously, we think about this for human standpoint, but the reality is a lot of this occurs in situations like breeding of animals, where people do this on purpose to get very specific traits because they want purebreds. They want to get rid of all the hybrid versions, which is the heterozygous. They want to have homozygous traits. So they'll do this in order to do it, but it will generate, over time, inbreeding issues. F equals 1 is homozygous from a common ancestor. So if you go along and you get an F value of 1, that means that is a direct link from one ancestor to the next ancestor. That could be like basically a parent to a child mating would cause that kind of concept. F0 means if you come along, they're not related at all. There's no common ancestor within a few generations. Anything past about 3 to 4 generations, it usually is okay, so they can kind of look at that as it's not close enough that it's going to cause a relation. The math will go down to near 0 very quickly after a few generations. So why is inbreeding so bad? You hear about it all the time. There must be a reason why. Obviously, we do it for purebred animals, so there must be a reason why it works for some event, but also for another event it becomes bad. Number one, it lowers fitness because it starts to get rid of very particular types of genotypes. And that lowers the fitness ability because it creates very specific groups. It decreases that genetic diversity because it will start to eliminate traits out of population. If you start having lots of inbreeding, all of a sudden one trait becomes more dominant than another, starts to show up more often, and you get rid of the other trait in the population. And if you ever change the population, all of a sudden that becomes bad. It also increases deleterious mutations, which means the chance of a bad mutation occurring where it's going to cause a negative fitness value is much higher, because as you keep making these copies over and over again with each other that are all related from the same original gene, it tends to interfere and cause more mutations to occur, a higher mutational rate. So that's why you tend to see a lot of these mutations occur in small populations that do inbreeding, where it shows up more rapidly and more quickly. We call that inbreeding depression because it starts to push down upon the fitness level of the population and cause it to basically become depressed and not be very effective, not be very robust. Self-fertilization is probably considered the worst form of inbreeding. Obviously with us, right now you can't do it naturally. You could do it artificially through things like in vitro fertilization and things like that, theoretically you could do that in the human population, but we typically have rules against that. But animals such as animals that can do asexual reproduction, that is a problem. If they are basically can reproduce themselves by doing sexual fertilization but use their own sperm and egg to mate with themselves, then that becomes a type of self-fertilization, which can become problematic. Plants do this, which can cause even problems for them as well. So it's an extreme form of inbreeding. Because in that case, it's 100%, it's always 1. F is always equal to 1. You're going to lose heterozygosity very quickly. You're going to lose all heterozygotes. You're going to end up with just either all homozygous dominant or homozygous recessive, all the A's and all the little a's, or one of them will go away completely. You may end up very quickly with a population of just all big A's or just all little a's, which means they have very to no diversity whatsoever. So you can see over time, these are your homozygotes and your heterozygotes. You start with a population where you have 100% heterozygotes. If you do inbreeding of values over time, what happens is it very quickly, within about 5 generations, goes almost completely to almost all homozygotes, no heterozygotes. They go away. And you can see here in the curve how these things end up fluctuating the curve. They basically flatten and drop down the percentages of heterozygotes here in the middle. They get rid of them. They go from here in the middle to just all of a sudden being dropped completely. From 100% to all of a sudden gone, and having 2 separate populations away from each other. Or could push all of them into one way and only have only that group there as well, or only this group here, to get rid of those kind of things.